So the topic is alcohol fermentation. It is also based on research work. First is abstract. The origin of modern fruits brought to microbial communities has an abundant source of rich food based on simple sugar. Yeast, especially Saccharomyces cerevisiae, usually become the predominant group in these. One of the most prominent and unique features, likely a winning trait of these yeast, is their ability to rapidly convert sugar to ethanol at both anaerobic and aerobic conditions. Why, when, and how did yeast remodel their carbon metabolism to be able to accumulate ethanol under aerobic condition at the expense of decreased biomass? So the recent data, carbon metabolism in these species, and attempt to reconstruct the ancient environment. So the first step towards so-called fermentative lifestyle was the exploration of anaerobic, resulting in increased metabolic capacity of degrade sugar to ethanol. The strength glycolytic flow had in parallel beneficial effect on the microbial competition and later evolved as new tool. The basic aerobic alcohol fermentation ability was subsequently upgraded in several lineages by evolving additional regulatory steps such as glucose repression. Tree effect, one of the most prominent features of baker's yeast, it is ability to rapidly convert sugars to ethanol and carbon dioxide at both anaerobic and aerobic condition. Under aerobic condition, respiration is possible with oxygen, the final electron acceptor, but it exhibits alcohol fermentation under the sugar reaches a low level. The phenomena is called Crabtree effect. The yeast expressly this trait called Crabtree positive yeast, in contrast, Crabtree negative yeast like fermentative products. However, it is possible to obtain pure respiratory utilization of glucose under aerobic condition if the glucose concentration is low, for example using a glucose. Then is However, the complexity of glucose repression regulatory network is far from being completely understood. Some of the regulatory activities operate the transcription level and some others operate directly. Different physiological and molecular approaches have been used as a background for the current definition of Crabtree effect. Then is ecological perspective and yeast lifestyle. Every autumn when fruits ripen, a fierce competition for the fruit sugar starts within the microbial communities. Yeast and its close relatives usually become the predominant group with freely available mono and polysaccharides. The fast sugar consumption, ethanol production, and tolerance, the availability to propagate without oxygen, are likely some of the winning traits responsible for the competition. At least three lanes, including budding and fission yeast, have apparently independently evolved the metabolic ability to produce ethanol in the presence of oxygen and excess of glucose. On the other hand, it can grow only poorly on ethanol as sole carbon source. In short, this life strategy is based on that yeast can consume very fast more sugar than the other species, convert it to ethanol to inhibit the growth of other species, especially bacteria, and then consume the remaining carbon once they have established competitive dominance in. Ability to grow anaerobically. The availability of oxygen varies among different species. One of the main problems an organism faces under anaerobic condition is lack of final electron acceptor. This completely eliminates the activity of Krebs cycle, respiratory chain, and mitochondrial ATP generation. As a response to hypoxic and anaerobic conditions, organisms have developed several processes to optimize the utilization of oxygen and even reduce the dependence or presence of oxygen. The ability of yeast to grow under oxygen limited conditions seems to be strictly dependent on the ability to perform alcohol fermentation. Well, we can say that, for example, for the amino acid synthetic pathways need to originate from modified and metabolic network. On the other hand, in yeast, some compounds such as unsaturated fatty acids and sterols cannot be synthesized in the cell under the anaerobis and must originate from the medium from previous and aerobic growth. So, how to deduce the yeast evolutionary history? The one of the yeast genomics has provided a tool to reconstruct several molecular events which have reshaped the budding yeast during evolutionary history. While the origin of more complex traits such as Crabtree effect is often not easy to determine, usually using on the genome analysis approach. Communities a new, larger, and increasingly source of food. On the other hand, ancient yeast could be hardly produce the same amount of new biomass as bacteria during the same time interval could therefore outcompeted. So the Crabtree effect, which is background for the yeast make accumulate consume strategy, results in lower biomass production. then just more glucose would be consumed in this case. What could be the original driving for that increase the flow through the glycopath? So the conclusion is this: during the recent years there have been more and more research focused on non-conventional yeast, especially many of these yeast got their genomes skewed. These sequenced data now help to deduce a reliable phylogenetic relation among yeast, provide us with the possibility to reveal evolution fingerprints which have remained preserved in the genome. These data now should be complemented, and we speculate that exploration of anaerobic and later on competition with other microorganisms were driving forces behind the remodeling of yeast carbon metabolism. So this was all about alcohol